Hello everyone and welcome to Lecture 6. We're going to talk about neurotransmitters and their receptors. We'll start off with the principal excitatory neurotransmitters, then the common inhibitory neurotransmitters. Then we'll talk about some neuromodulatory transmitters that use G protein coupled receptors to carry out their action. And we'll finish off by talking about some unconventional transmitters that aren't stored and released in the normal vesicular manner. Okay, the excitatory transmitters for the most part are going to be glutamate and acetylcholine. Glutamate is going to be the principal excitatory neurotransmitter in our central nervous system. Acetylcholine will be the principal excitatory neurotransmitter outside of our central nervous system. So acetylcholine is going to be synthesized from the acetyl group from acetyl coenzyme A, which hopefully you recognize from your Krebs cycle, and a choline molecule. The enzyme that carries this out is choline acetyltransferase, or CHAT, and the enzyme name tells you exactly what it does. It's going to transfer an acetyl group onto choline, hence the name choline acetyltransferase, and that will create the neurotransmitter acetylcholine. Because acetyl-CoA is a part of the Krebs cycle, it has to be transported from the mitochondria, and so this action is the slowest portion. This is what determines the rate at which we can synthesize acetylcholine. Acetylcholine is going to be packaged into vesicles and then released as discussed in our last lecture. Acetylcholine is going to bind to two types of receptors. The nicotinic receptors are the ionotropic receptors, which we'll talk about now, but you should keep in mind that there are also muscarinic acetylcholine receptors, which are G protein coupled receptors. So they're not ion channels, but the nicotinic ones are. So we'll focus on them here. You're going to find nicotinic acetylcholine receptors in our muscle cells, as well as in neurons. Mostly you're going to find these presynaptic. They're going to have a modulatory function. In the peripheral nervous system, you'll find the nicotinic acetylcholine receptors at the neuromuscular junction, so on muscle cells, where it acts as your standard postsynaptic excitatory receptor. There are a number of different subunits that can make up a nicotinic acetylcholine receptor. You're not going to be expected to know the different types at the electrical organ versus the neuromuscular junction, and hell, we don't have electrical organs, so it doesn't really matter that much. You might want to know that alpha subunits are important because they have the ligand binding site. So without the alpha subunit, you can't bind acetylcholine, and if you can't bind acetylcholine, you're not much of a receptor. The nicotinic acetylcholine receptors, although they can be made of a number of different subunits, they're always made of 5 of them. They're always pentamers. As you can see in this cartoon on the bottom or middle, you can see that there's 5 different subunits that make up nicotinic acetylcholine receptor, and each of these subunits has 4 transmembrane domains in them. The one that's really important to us would be the second. You'll notice that it creates the pore. If you look on the bottom, you'll notice that the ion pore is lined with the green second transmembrane domain. So it's the second transmembrane domain that must move in order to open the ion channel. So once acetylcholine binds to the receptor, it causes the second transmembrane domain to rotate. Under ordinary conditions, as we can see in portion B of this illustration, the second transmembrane domain is kind of kinked inwards, and it has a number of leucine amino acids that it lines up to create this hydrophobic ring in the center that restricts the movement of water and thus ions. Upon the binding of acetylcholine, all of these transmembrane domains rotate. That's what we see in part C. So they rotate out of the way, kind of like an aperture in a camera rotates to open up. When it rotates, it moves the hydrophobic leucines out of the way and instead lines the ion pore with polar amino acids so that water and cations can move through the ion pore. And that's what allows the nicotinic acetylcholine receptor to be an excitatory neurotransmitter receptor, because it's a non-specific cation channel, so it's going to reverse at around 0 millivolts, which is more than sufficient to drive action potentials in neurons and muscles. There are a few drugs that act on nicotinic acetylcholine receptors. Principal among them would be nicotine. That's where it gets its name. Nicotine is an agonist for the nicotinic acetylcholine receptors. If you were to apply nicotine and record, you'd find that you get an excitatory current in response to nicotine. It, of course, quickly desensitizes, as most all receptors do, and you can block it. So if you were to block the nicotinic acetylcholine receptor, you see no response to nicotine, telling you that nicotine is indeed activating nicotinic acetylcholine receptors. There are also many venoms that are going to act on the nicotinic acetylcholine receptor. They'll be competitive antagonists, so they're going to compete at the acetylcholine binding site. For example, alpha bungarotoxin and MLA shown here. You can see they're decreasing the postsynaptic response to nicotine to stimulation of the motor neurons. So let's look at the graph on the right. The open, what looks like circles to me on top, that's your control. This is your nicotinic acetylcholine receptor current amplitude under normal conditions. So here they're recording for about 30 to 35 minutes, and you can see that we're getting about 100 picoamps, between 100 and 120, fairly reliably. So that's what we'll consider normal here. By applying these different toxins, like alpha bungarotoxin, that would be the open, perhaps, circles. I don't know. The one that falls, it's fairly small on my screen. It's also labeled alpha BGTX. That's going to prevent the activation of nicotinic acetylcholine receptors. These toxins are going to, of course, cause paralysis because nicotinic acetylcholine receptors are responsible for causing muscle contraction. And so any antagonist to the nicotinic acetylcholine receptors is going to thus cause transmitter. We also have glutamate. That's an excitatory amino acid transmitter. Glutamate and GABA are closely linked. Glutamate's the excitatory transmitter. GABA is the inhibitory transmitter, and they can be made from one another. GABA is made directly from glutamate, and then glutamate can be synthesized in a very roundabout way from GABA in what we call the GABA shunt. So glutamate is going to be in every single cell in your body. Glutamate is a normal amino acid that we use to build proteins. So every cell has it, and they make it from a few sources. Since we don't transport glutamate across the blood-brain barrier, we have to synthesize it from either glutamine or alpha-ketoglutarate. Glutamine is used to make glutamate in a single-step reaction. The enzyme glutaminase will convert glutamine to glutamate. There's also GABA transaminase, which can be used to convert alpha-ketoglutarate to glutamate. So alpha-ketoglutarate is a component of our Krebs cycle. So this is an important metabolic substrate. So it's not ideal to take away alpha-ketoglutarate to make glutamate. So principally, we'd prefer to use glutamine to make glutamate and preserve our alpha-ketoglutarate so we can continue to make ATP. But when times get tough, we can dip into that. It is an option. Once glutamate has been made, glutamic acid decarboxylase, or GAD, is going to convert it into GABA if neurons synthesize GABA. But more on GABA later. Here we'll focus on glutamate. There are many different types of glutamate receptors. Of the ionotropic receptors, there are two broad categories. There's the non-NMDA, which includes AMPA and kainate receptors, and then there's the NMDA receptors. Non-NMDA receptors are strict ligand-gated ion channels. Some of them will be calcium impermeable if they contain the GLU-A2 subunit, but other than that, they're non-specific cation channels. When glutamate binds, they open, cations move across the membrane and bring the membrane potential toward 0 millivolts. So they cause depolarization. AMPA receptors and kainate receptors are going to be tetramers of different subunits, but they, in the end, are ionotropic glutamate receptors. When glutamate binds, cations will cause depolarization. NMDA receptors, on the other hand, while they are ligand-gated, they are also voltage-gated. by way of the magnesium block. So NMDA receptors are made of many different subunits, which I don't care if you know. They need to have GLU-N1 and GLU-N2. GLU-N1 is required for pore formation, and GLU-N2 has the glutamate binding site. Both of those are necessary. But then they'll have a mix of other different subunits to give them different properties, which we won't really be discussing in this class. So glutamate is going to bind to the NMDA receptor. So must glycine, it turns out. So this inhibitory neurotransmitter is actually also excitatory by way of the NMDA receptor. So NMDA receptors will bind glutamate and glycine, but nothing will happen unless the cell is first depolarized. And that's because of that magnesium ion sitting in the middle of our NMDA receptor. In order for us to actually allow the ions to move through, so if we're going to get calcium and sodium to enter and potassium to leave, we have to get that magnesium out of the way. This is what we call the magnesium block. And that's why at resting potentials we have very low NMDA receptor currents. It's because of the magnesium block. We have to first depolarize a bit. That positive membrane potential is going to repel magnesium and push it out of the ion channel, and thus allow the other cations to move through the membrane. So what we're looking at in this graph would be the IV relationship, or the current and potential relationship. The membrane potential is shown on the x-axis, rest is on the left, and then more depolarizing potentials are to the right. You'll notice a J-shaped curve for the NMDA receptor, shown in squares there. AMPA receptors are linear because they don't have the magnesium block. So at negative potentials, cations flow in. There's not a magnesium sitting in the way. You can get a robust current. The current is shown on the y-axis. But at negative potentials, like at minus 80 there, you'll notice that the NMDA current is very, very low. It's near zero. And that's because of the magnesium block. This makes the NMDA receptor a molecular coincidence detector because it's only active if glutamate and glycine are binding and the neuron has been previously depolarized. The importance of this will come up whenever we talk about learning. This is very, very important. And we want to restrict the activity of NMDA receptors because they don't desensitize as rapidly as AMPA receptors. When you compare the amount of ion flux with AMPA and NMDA receptors, you can see AMPA receptors activate very quickly, but then they also desensitize rapidly. So you get a very brief current. NMDA receptors open more slowly, but they're active for much, much longer. This is analogous to the voltage-gated sodium and potassium channels, where sodium channels open quickly, but they inactivate. Potassium channels open more slowly, but they remain active until we have repolarization and eventually hyperpolarization. This is similar. AMPA receptors open immediately and then desensitize. NMDA receptors open a little more slowly, but they remain open for much, much longer. That means that they can move a much greater amount of ions. And principal among these ions would be calcium. NMDA receptors are much more permeable to calcium than AMPA receptors in general, and because they're open much more longer and more calcium permeable, they're going to allow a greater calcium influx. Calcium is a very important secondary messenger, which we will talk about as we discuss intracellular signaling pathways. But that's what allows NMDA receptors to kick off some long-lived changes in neuronal function. So NMDA receptors are coincidence detectors that detect when two things happen together, and then because of the amount of calcium that they let come into the cell, they're able to change how that cell functions as a result of experience. Alright, again, we're going to touch on this when we discuss learning. For now, we should just understand that AMPA receptors mediate fast excitatory transmission. NMDA receptors are coincidence detectors that allow the neuron to change in response to experience. Okay, so we got our excitatory transmitters. Now let's move on to our inhibitory transmitters. We're not going to discuss glycine. You should know that glycine is, for the most part, an inhibitory transmitter, except when it acts on the NMDA receptor. But the receptors for glycine and GABA are very, very similar. So we'll talk about just GABA here. And we're going to focus on GABA-A receptors because those are the ionotropic receptors. Keep in mind there are also GABA-B receptors, which are metabotropic. But here we'll focus on the ionotropic receptors. GABA A receptors are going to allow chloride to move across the membrane, and so they are going to be inhibitory receptors. They're made of a variety of subunits. Again, you won't need to know them. You should just know that there are many of them. And the different subunits are going to create different kinetics and drug specificity for the different GABA receptors. The GABA receptors are going to be pentamers of their different subunits, and the ligand binding site is between an alpha and a beta subunit, so you need at least an alpha and a beta. Just like all other receptors, the second transmembrane domain will line the pore. In this pore, rather than allowing cations to move across like the other receptors we've talked about, now we're letting chloride move through. All right, and so chloride is going to slightly hyperpolarize, but mainly shunt excitatory input. There are a few drugs that are going to act on GABA A receptors. The benzodiazepines and barbiturates are going to affect the frequency of channel opening and the open time of the channel, respectively. Ethanol appears to do both of these effects. Okay, so if we look at the benzodiazepine diazepam, we can see that it's going to act as a GABA A agonist. It's going to increase the amount of GABA A current. So if we look at the top illustration, we can see the postsynaptic response to brief applications of 20 micromolar GABA. And so you can see that we have a brief but sustained inward current as long as GABA is around that quickly reverses. When the same concentration of GABA is applied with this benzodiazepine, you can see that the same amount of GABA is able to elicit a much, much greater current. Okay, and so the benzodiazepines, barbiturates, and ethanol are going to thus inhibit the activity of neurons by acting as GABA A agonists. There are also antagonists that function as pore blockers, such as picotoxin. So picotoxin is going to fit into the pore. If we look on the bottom here, the mutations that interfere with picotoxin binding are the ones that occur in the pore. So they looked at 3 different mutants. They called them alpha, beta, and gamma. So just essentially A, B, and, well, G, but gamma is the 3rd letter. So 1, 2, 3. Mutants 1, 2, and 3 all have mutations in the ion pore, and you can see that the concentration of picotoxin, shown on the x-axis, must be greater in order to sufficiently inhibit GABA currents. Okay, so there are GABA agonists, there are GABA antagonists. GABA antagonists tend to cause problems because they can lead to hyperexcitability of neurons and create excitotoxicity. The GABA agonists tend to reduce inhibitions and worries, so they'll be used for anti-anxiety medications, and sometimes people will self-prescribe ethanol to treat their anxiety. Those are the main ionotropic receptors that we need to be aware of. But there's a wide variety of neuromodulatory transmitters and even the same transmitters we just discussed that act on G protein coupled receptors that are going to change cell function over very long time courses. We'll talk about the downstream signaling in more detail in other lectures, but every neurotransmitter is going to have a metabotropic receptor or 2 that it acts on. GABA A receptors are ionotropic, but there's GABA B that's metabotropic. Glutamate acts on AMPA kainate or NMDA receptors, but there's also a variety of different mGluRs that are divided into 3 different classes. Acetylcholine, it acts on nicotinic receptors, but it also acts on muscarinic acetylcholine receptors. The different neurotransmitters we're going to bring up, so dopamine, norepinephrine, serotonin, histamine, purines, peptides, those all act on metabotropic receptors. Okay, metabotropic receptors are going to change cell function over long time courses. They're not going to mediate fast ion currents. Instead, they're going to modify cellular signaling. They're going to do that by the way of G proteins. G proteins are going to be discussed in greater detail in subsequent lectures, but you should understand that a G protein is a protein that binds a guanine nucleotide And depending on whether it's binding GTP or GDP, it's going to either be active or inactive. And this G protein will act on some target within the cell. So a G protein coupled receptor is a receptor that's bound to a G protein. Every G protein coupled receptor has 7 transmembrane domains. This is a typical feature of all of them. The ligand binding site is typically buried in the membrane, although not always, but typically it is. And in the intracellular portion, which we can see on the bottom of this illustration, we have the binding site for our G protein. There are a few different types of G proteins, and we'll go through them later. You should just be aware that G proteins are the intracellular protein that translates ligand binding to some change in cellular activity. Okay, so let's talk about a different class of neurotransmitters, the monoaminergic transmitters. These are going to all be built from different amino acids. We're going to either start with tyrosine or tryptophan, whether we're talking about the catecholamines or serotonin. So with catecholamines, it starts with tyrosine. The enzyme tyrosine hydroxylase will modify tyrosine to create L-DOPA. It's going to do so by adding a hydroxyl group onto tyrosine. Tryptophan will be modified by tryptophan hydroxylase. Again, we're adding a hydroxyl group onto tryptophan to make 5-hydroxytryptophan. These two intermediates are then acted on by the same enzyme to create dopamine or serotonin. So L-aromatic amino acid decarboxylase is going to then convert L-DOPA to dopamine, or 5-hydroxytryptophan to serotonin. We're done with serotonin at this point. We are not done as far as synthesizing catecholamines. So dopamine will be further processed to create norepinephrine. This is done inside of synaptic vesicles, which is kind of unique. So norepinephrine is constructed within synaptic vesicles. Epinephrine, which is made from norepinephrine, must be made after norepinephrine is created, secreted, and then taken back up. The norepinephrine is then used to create epinephrine. Epinephrine is largely absent in the central nervous system. There are very few neurons that make it, so we can forget about epinephrine to some degree. This acts more peripherally than centrally. Norepinephrine, we do see many projections within the central nervous system. But these are just a variety of single-step reactions where we're taking some precursor and modifying it. It's just stepwise modifications to create L-DOPA from tyrosine, to create dopamine from L-DOPA, to create norepinephrine from dopamine, and then epinephrine from norepinephrine. Just as we have enzymes to create our monoamines, we also have enzymes to break down our monoamines. Monoamine oxidase is going to break down our monoamines, and then catechol O-methyltransferase is going to also break down our monoamines. They'll do so in different ways. Catechol O-methyltransferase is going to add a methyl group onto the oxygens that are circled in red here. So you can see that the different monoamines are going to be modified at pretty much the same location. Monoamine oxidase is going to replace the nitrogenous groups that are circled in blue with a hydroxyl. By doing this, these enzymes are going to modify the structure of the neurotransmitter, and thus they're going to prevent it from binding with its receptor. The inhibitors to monoamine oxidase used to be used to treat depression, but because monoamine oxidase acts on so many different transmitters, it's kind of a messy drug, and we've since moved on to other treatments. But the idea was to prevent degradation of serotonin and other monoamines that will boost their levels and thus improve our mood. But what you find is that these monoamines have widespread connections throughout the brain, and they have a variety of functions that they're involved in. And so drugs like MAO inhibitors can have some pretty nasty side effects. Norepinephrine is going to arise largely from the locus coeruleus, and then it's going to be released throughout the brain. So we don't have every neuron in the brain synthesizing norepinephrine. It's really just a small group of neurons. that then project throughout the brain, and they're going to help control our attentiveness, some behaviors, particularly sexual behaviors and appetite. More on that potentially later in the class. But norepinephrine, like many of the neuromodulatory transmitters, are going to be synthesized in very discrete locations in the brain. Glutamate is made essentially throughout the entire brain, as is GABA. So these are widely used neurotransmitters. The neuromodulatory transmitters are going to come from specific locations. For example, dopamine is going to be synthesized mostly in the substantia nigra, but also in the ventral tegmental area. And then those two regions are going to project in three different pathways to mediate different activities. So we'll control our movements to some degree by the nigrostriatal system, so from the substantia nigra into the striatum, into our basal ganglia. So that's going to allow dopamine to regulate movement. We'll see this is very important in Parkinson's disease, but dopamine does other things as well. The ventral tegmental area has two projections. There's the mesolimbic system, which projects to our limbic system. That'll help regulate our emotions, the feelings of reward, and addictive behaviors that are associated with using drugs that affect dopamine function. Then there's the mesocortical system, where we project from the ventral tegmental area to the prefrontal cortex, and this is going to regulate our short-term memory, our planning, so this will help control our actions as well. Now, dopamine is going to act on two different types of receptors. Some of them are going to stimulate, some of them are going to inhibit. So the D1 class stimulate, the D2 class inhibit. Here's the activity of D2 class receptors. So we're looking at the pacemaking activity of a dopamine neuron. And when we apply dopamine to that dopamine neuron, in the top portion of this illustration, we can see that the action potentials, shown as vertical lines, stop. So we apply a brief burst of 30 micromolar dopamine, and you can see that the action potentials cease. Then they come back after the dopamine has washed out. This is because of the activity of D2 dopamine receptors. Because if we inhibit a specific D2 class agonist, quinpirole, that's what we see in the bottom, we can also block the firing of action potentials. Now these are G protein coupled receptors, and they're going to be coupled to two different types of G proteins. The D1 class are going to be coupled to GS type G proteins that are going to stimulate adenylyl cyclase. D2 class will be coupled to GI or GO, which will inhibit adenylate cyclase. So D1 is activating adenylate cyclase, D2 is inhibiting adenylate cyclase. Adenylate cyclase is an enzyme that creates cyclic nucleotides. So it's going to convert ATP to cyclic AMP. Cyclic nucleotides are going to bind to certain cation channels and cause them to open. So the higher the level of cyclic AMP, the more cation permeability you have, and thus the more depolarization you experience. So by activating D2 dopamine receptors, we inhibit adenylate cyclase, lower cyclic AMP levels, and thus reduce our cation permeability. So our membrane potential moves away from the reversal for nonspecific cation channels. In other words, it hyperpolarizes. The opposite happens with D1. Okay, more on adenylate cyclase later on, but this is just a taste of things to come. Here we have two different dopamine receptors. They're both G protein coupled receptors, but because they're coupled to different G proteins, they have different effects on cell function. This takes some time. All right, you'll notice that the cell doesn't stop firing immediately when dopamine is applied. There's a brief delay because the drug has to wash on, and then the receptors have to be active, and we have to modify the activity of an enzyme. So it's a little bit slower than the ionotropic signaling that we're used to seeing thus far. Serotonin, much like the other monoamines, is going to be created in our brainstem and midbrain, and then sent throughout our central nervous system. So the raphe nucleus is what's going to generate serotonin in this case. And then it's going to project to essentially all parts of the brain to regulate our mood, eating, sleep, arousal, and pain. Most of the serotonin receptors are going to be metabotropic. So they're going to have different effects on adenylyl cyclase. There's also another enzyme that can be regulated, phospholipase C, depending on which serotonin receptor we're talking about. Some are going to be found postsynaptically, some will be presynaptic, and then there are some that are actually ionotropic. They're going to be cation channels that mediate fast synaptic signaling, but most are going to be metabotropic in this case. Depending on their coupling, they're going to have different effects on neurons. Some can stimulate, some can inhibit, just like dopamine. Histamine is another important transmitter. This is going to be synthesized in again, one location in the brain. This is in a slightly higher region of the brain. This is in the hypothalamus, in the tuberomammillary nucleus. Then histamine is going to be released throughout the brain. Histamine is, of course, synthesized from histidine by histidine decarboxylase, and then released widely throughout the central nervous system to create feelings of wakefulness. So depending on which receptor histamine binds to, it can also either stimulate or inhibit neuronal activity. The H1 histamine receptors clearly increase nerve cell activity. So when we apply histamine, we can see that there's an increase in the frequency of action potentials here. So in panel A on top, you can see control, where we don't have any histamine applied, just some receptor blockers. We've blocked the inhibitory receptors. Now when we apply histamine, the inhibitory receptors are blocked, so only H1 gets activated, and we get an increase in the firing rate. This is summarized in panel B. The y-axis is showing you the firing rate frequency. 100% means it's normal, that's baseline. After we apply histamine around the 10-minute mark, you can see an increase in the frequency of action potentials, and this is sustained until the H1 blocker is applied at around the 22-minute mark or so. So tripolidine is going to block H1 receptors, and so that excitatory drive is lost, and our firing rate returns to normal. Now, because histamine tends to create feelings of wakeness, antihistamines tend to make us feel drowsy. The newer non-drowsy versions are actually just blood-brain barrier impermeable, so that they will act in the periphery to reduce our allergic response, but they won't inhibit the wakefulness feeling that histamine creates in our central nervous system. Adenosine is another neuromodulatory transmitter. This is not made from amino acids. This is a derivative of nucleic acids. So ATP is going to be found in synaptic vesicles, and so when neurotransmitters are released, generally they also release some ATP. That ATP is rapidly broken down into adenosine, which will bind to adenosine receptors. In general, adenosine is going to cause feelings of drowsiness, so it's the opposite of histamine. That means that adenosine antagonists like caffeine are going to block that drowsiness and thus make us feel more awake. There are different types of adenosine receptors which do have different coupling. Some of them are going to be GS, some of them will be GI, so they're going to have different effects on adenylyl cyclase, just like dopamine receptors. Now there are, of course, peptide transmitters. So neuropeptides are a very diverse group of neurotransmitters. These are fairly costly to make because we can't recycle them like we do our classical transmitters. Instead, we have to synthesize them de novo every time we want them. And only very strong levels of neural activity will induce their release. But just like the other neuromodulatory transmitters, when they are released, they will activate the G protein coupled receptors and cause some long-lived change in nerve cell function. A great example of this would be enkephalins, which bind to opioid receptors and regulate our sensation of pain. We're going to have opioid receptors on the nociceptors that pick up noxious stimuli like tissue damage or intense heat. There will be opioid receptors on the nerve cells that then transduce that signal from the spinal cord into the central nervous system. And so the release of enkephalins is going to stimulate opioid receptors and tone down that signaling. So they're going to have a general inhibitory effect on the signaling between nociceptors and our central nervous system. And this is why we'll use opiates to help reduce pain. One of the problems with using opiates is that opioid receptors are also very much involved in our feelings of reward. So these drugs can be highly addictive because of that response. So the pain modulation happens mostly at the level of the nociceptor, but then acting in our central nervous system, in the limbic system and the basal ganglia, this can create reward responses and then thus addictive behaviors. Okay, the last transmitters that we're going to talk about here will be the unconventional transmitters. These are not stored in vesicles. They're also not released from the axon. These are going to be, for the most part, released from dendrites and cell bodies. So they're going to accomplish non-vesicular retrograde signaling. In other words, they're going to move backwards. So it's not axon to dendrite. In this case, it'll be dendrite to axon, for example. The first group would be the endocannabinoids. These are derived from lipids. So we'll take the phosphoinositides that are in our plasma membrane, and there's going to be an enzyme phospholipase C. This is a target of G protein coupled receptors. This is going to cleave them to create diacylglycerol and inositol phosphate. Inositol phosphate is really useful, but we can forget about it for right now. That doesn't matter here. Instead, diacylglycerol is then further cleaved by diacylglycerol lipase, and that's going to create the endocannabinoid 2-AG. Okay, 2-arachidonylglycerol. This is going to then freely move across the membrane because it's very much hydrophobic. But now it's no longer embedded in the membrane. DGL has cut out 2-AG, so now it's a free-floating hydrophobic molecule that can move through the dendritic membrane and then bind to the cannabinoid receptors on the presynaptic terminal. And that's what we're looking at here. So because of the lipophilic nature of endocannabinoids, we can't store them in vesicles. So that means anytime they're created, they then leave and carry out their signaling. So they need to be synthesized only on demand. They're going to bind to those cannabinoid receptors. These are G protein coupled receptors that are going to activate a number of different signaling cascades that in general reduce the release probability at the presynaptic site. In other words, the generation of endocannabinoids is going to decrease the likelihood that the presynaptic membrane releases more neurotransmitter. We generally create endocannabinoids when the postsynaptic receptor has been strongly activated. So when the cell gets activated strongly, it releases 2-AG to then tone down the activity of the presynaptic cell. This is just a homeostatic response. If the presynaptic cell is stimulating the postsynaptic cell at a very high level, endocannabinoids turn that down to avoid excitotoxicity. And they do that by binding to cannabinoid receptors. CB1 is the most common one. There's also CB2, but these are G protein coupled receptors that then act through a variety of signaling cascades to, in general, inhibit voltage-gated calcium channels. So we don't get as much calcium influx at the presynaptic site. Thus, we're not releasing as much neurotransmitter. They also inhibit adenylyl cyclase. Remember, that's the enzyme that creates cyclic AMP. By decreasing cyclic AMP levels, the cyclic nucleotide-gated cation channels in the presynaptic terminal are less active, and thus there's less depolarization in the presynaptic terminal. Thus, it gets harder to activate voltage-gated calcium channels. As a result of this, cannabinoids decrease presynaptic release. That's what their function is. HU210 is a synthetic cannabinoid. So this is not 2-AG. You can see the excitatory postsynaptic current amplitude drops after the application of this cannabinoid. And that's because of the activity of CB1 receptors. If we apply the CB1 receptor, SR, then that blocks the inhibitory effect of HU210. So on the bottom graph, what we can see is that the excitatory postsynaptic current amplitude stays relatively stable, even though the cannabinoid has been added. That's because the CB1 receptor has been blocked. This is showing you that the cannabinoids are indeed acting through the CB1 receptor and not some alternative signaling pathway. After they're released, cannabinoids are going to be broken down. Since we can't really reuptake cannabinoids because they freely move through the membrane, our only option here is to break them down. So 2-AG is going to be degraded by monoacylglycerol lipase, so MGL there. And there are a number of other cannabinoids and other enzymes that regulate them, but that's the basics of it. We take a membrane that's embedded in we take a phospholipid that's embedded in the membrane, that's right at the top there, PIP2. We're going to break it down into diacylglycerol, then we're going to turn that into an endocannabinoid, so 2-AG, and then we're going to break that down into an inactive derivative. So we have our endocannabinoids that are retrograde inhibitors. We also have nitric oxide, which is actually a retrograde stimulator. Nitric oxide is a small gas that's made from arginine by the enzyme nitric oxide synthase. Nitric oxide, because it's a small gas, can readily diffuse through membranes, and so it just diffuses in all directions and modifies the activity of nearby cells. There's no vesicular transport, there's no vesicular release. There's also no enzymes to break it down. It's just a small gas, so it will spontaneously decay into nitrate after about 30 seconds. The purpose of nitric oxide is to stimulate neurotransmitters by acting on voltage-gated calcium channels and stimulating the snare complex to form. As a result, nitric oxide is going to be able to stimulate neurotransmitter release. Here we're looking at the amount of acetylcholine being released. NLA is a neuronal nitric oxide synthase antagonist. When NLA is applied, you can see that there's a decrease in the amount of acetylcholine released. The open circles are showing you baseline, where there's no drug. They just recorded for 160 minutes and didn't do anything. The black squares, perhaps, are showing you the conditions where they have added drugs. So when they added NLA, you can see a very noticeable decrease in the amount of acetylcholine released. On the other hand, when they added a compound that released nitric oxide, you'll see a very rapid and noticeable increase in the amount of acetylcholine release. So again, this is an on-demand signal. We can't store it. So whenever neurons want to release nitric oxide, they have to make it, and then it's going to diffuse in all directions and stimulate the release of neurotransmitter. Alright, that does it for Lecture 6. I hope you'll email me any questions you have on the material, and I look forward to seeing you in the next lecture.